I'm Matthew Bio. I'm the Chief Science Officer of Cambrex, and I'm based out of the Snapdragon site in Waltham, Massachusetts. It's taking them a long time to ramp up, and it's very expensive, and it's dedicated machinery that does this solid phase synthesis that they're using in the manufacture. By moving to a liquid phase approach, you debottleneck that whole process of getting these peptides because you're not tied to a specialized reactor anymore. You're now able to leverage reactors that are all over the world at various scales up to tens of thousands of liters. We've seen this with mRNA and other things as well. When these modalities get proven so dramatically in the marketplace, people look to them for a lot of other things now. So, for example, there's a lot of these smaller peptides now that are being evaluated for cancers to stop protein-protein interactions, which is a really difficult target. And these peptides have enough of a size and space and surface area and enough different points on it that you can tweak very easily, that you can now go after these protein-protein interactions that were thought to be undruggable in some cases with these peptides. So the opportunity there is to help serve those clients, get their molecules to market using this technology, which is actually a much smaller footprint for them. It allows them to go faster to market because it utilizes some traditional manufacturing technologies. Yeah, so over the last couple of years, we've become really interested in this area of peptide synthesis. And I think we were able to identify a niche where we make small fragments of peptides and then we put those together, and we can do that entire sequence in liquid phase. Solid phase still has its place. It's very fast. It's useful in early development, and so we do that in early development. But as you move closer and closer to manufacturing, you want a sustainable process. You want something that's not going to cost a huge amount of in cost of goods driven by the use of excess solvent, reagents, and things like that. We're able to move to much more environmentally sustainable solvents, some that are plant-derived, like 2-methyl-THF, as opposed to petroleum-derived or halogenated, which are challenging for the environment. So this technology really fits very well with a sustainable footprint that pharmaceutical companies are trying to move towards in their manufacturing. So really there's 3 sites that are engaged in this liquid phase peptide technology development. The first is Snapdragon. So at the Snapdragon site, we're actually developing the chemical technology to enable us to keep these things in solution, to be able to wash away the impurities, and to be able to do that in an iterative fashion to build up the fragments that we need. And then we're studying how to bring those fragments together such that they don't cause new impurities. So when we want to build a 40-mer peptide, we're going to build that up from 4 10-mers maybe. We need to know where to divide those so that they don't cause new impurities from epimerization or whatnot to form. And so all of that technology and the way that we address that technology in the manufacture, what does the equipment train look like? We're using membrane technologies like nanofiltration and tangential flow filtration to purify these molecules, which is somewhat unique to synthetic chemistry. So that's the piece that we're delivering. How are we actually going to make these? There's 2 other critical factors. I mentioned one, Edinburgh, where they're looking at, okay, once we've made that peptide, how can we crystallize it to high purity and to a stable form? They're using their expertise in solid phase characterization and in phase diagrams to really understand how are we going to get a crystalline form out of this that provides purification, stability, and really just you're not doing these very energy intensive things like trying to remove water from a peptide. If you're isolating in water, we don't have to go through that really energy intensive isolation process if we can instead develop a crystallization. So Edinburgh is doing that part. And then our colleagues down in Durham are looking at high resolution mass spec as a way to characterize all the impurities that could possibly be in there. And that's really important because peptides are these they're complex molecules that have multiple stereocenters and functional groups throughout them, and each one of those could be the source of a new impurity. And so to study that impurity profile, you really need some sophisticated high-resolution mass spec equipment, and also to be able to do peptide digest so that you can sequence the peptide and be sure that you have the right sequence that you've made. So the three groups together are able to work collaboratively to deliver what at the end is really what the client wants, is an end-to-end. We just need the peptide therapeutic out at the end in a way that is quality controlled, available on scale, and in a shortest timeline.